In this video, we are going to look at what biotechnology is and how we can use genetics to help treat disease. So biotechnology is using our understanding of biology and genetics to improve human quality of life. And we can do this in a number of ways, including healthcare, agriculture, and protecting and improving the environment. So remember, when we talk about biotechnology, the term genome represents all the genes that an organism has. And then within that genome, there is the DNA sequence. So that's the particular set of A's, T's, C's, and G's. And this is going to be unique for each person. And the particular DNA sequence is going to help determine your individual traits, but it can also influence your risk of disease. The Human Genome Project is over 30 years old at this point, but what it did was it helped us actually for the first time sequence the human genome. In other words, determine the code for humans. What are all the human genes that make us? And then from that, we have been able to learn how to actually sequence people's DNA. So we can actually determine a person's unique genetics by looking at the actual code, the A's, T's, C's, and G's for individual people. So let's look at how we can actually apply this to healthcare. So it turns out that most medical issues have some genetic influence. And your particular genetics can influence how likely it is for you to get certain diseases, how fast those diseases progress and how severe they are, and how you respond to treatments. So when we're diagnosing and treating illness, we can actually use DNA technology to help identify the genetic causes of that disease. That can be both in the patient, so what do you have genetically that is helping to increase your risk or cause you to have this disease? But it can also be with the pathogen. So what is the genetics of the virus or the bacteria that is actually causing your disease? And with that information, we can potentially create treatments that are specific to those genetics. In other words, we can create medicines that are unique to each patient and potentially vaccines that are unique to each specific pathogen. In addition to creating medicines that are specific to genetics, we can actually use things like bacteria to make proteins that we might need, like, for example, insulin. That's something that diabetics need. And what we can actually do is create what's called a bacterial transformation, where we insert the human gene for insulin into a bacteria and create recombinant DNA. In other words, I'm combining DNA from both the human and the bacteria, and then I can make the bacteria create the insulin. So remember that DNA codes for RNA, codes for proteins. So if I add the genes to an organism's genome, it'll make the protein. So when I combine the DNA from the bacteria and the human, I can get the bacteria to create those human proteins because it has the human genes. So let's look at how that might work. So on a human chromosome, you have a gene for insulin. So you have that DNA sequence. I can use these fancy enzymes called restriction enzymes, which work kind of like scissors, to cut that particular DNA sequence out. And once I have that, then I can actually do something with it. So remember, bacteria, in addition to their normal DNA, also have plasmids, which are small bits of DNA that can be passed between bacteria. So what I'm going to do is take that same restriction enzyme and I'm going to cut the plasmid. So now I can actually insert this human gene into the plasmid. So now it goes bacterial DNA, then section of human DNA, and then bacterial DNA again. If I can get that bacteria to take this plasmid in, I have now transformed that bacteria, and that bacteria can use its enzymes and ribosomes to transcribe and translate that gene into the human protein. So then I actually have these bacteria making human insulin, which you can bottle up and send out to diabetics. Now, another thing that we can use genetics to do is to assess risk, because your disease risk is influenced by your genetics. So we can use your particular genetics to identify your risk of particular diseases and then work to either prevent or reduce them through things like diet, exercise, medicines, etc. Finally, in the future, we may be able to actually cure many of these genetic diseases through something called gene therapy. So if you have a bad copy of a gene that is causing you to have a high risk or have a disease, we can potentially go in and replace that gene with a good copy, which would cure your disease or reduce your risk to the normal levels.